welcome to the latest edition of Chet Chats, Union's online luncheon series. Chet Chats connects our faculty with alumni, parents, and friends for serious thought and discussion. I'm Phil Wida. I work in the communications department at the college. I'm pleased to introduce today's presenter. Scott Kirkton is an associate professor in the Department of Biological Sciences. He earned his Bachelor of Science degree from Denison University and his PhD from Arizona State University. He was also a National Institutes of Health postdoctoral researcher at the University of California, San Diego Medical School. Professor Kirkton joined Union in 2006. Professor Kirkton is an animal physiologist. He and his research students investigate fundamental questions about how changes during an animal's life history impact biological processes. Specifically, they use insect models to examine how development alters the biochemistry, morphology, physiology, and biomechanics of respiration and locomotion. Following his presentation, Professor Kirkton will entertain questions submitted by you, the viewers. You can email your questions at any point to alumni@union.edu. Again, that's alumni@union.edu. And now it's my honor to introduce you to Professor Scott Kirkton.
Thank you, Phil. I'm excited to share some of our lab's research with you today. When I tell people that I'm an entomologist, somebody that studies insects, the first question I'm asked is, was I that boy crawling around in the ground with a magnifying glass looking for bugs? And I wasn't. In fact, I was more likely to be playing Atari or riding my bicycle. I was so scared of bugs that in 5th grade, when we had to do a bug collection, my mom had to do it for me. This is very different than my daughters, who are very interested in insects, and my oldest actually has 3 pet Madagascar hissing cockroaches. So how does one go from being afraid of insects to making it their career, studying bugs? So that happened as an undergrad. I attended Denison University in Ohio, and as a sophomore, the only biology course that I could get into was entomology. I was hesitant about taking it, but I needed a biology course so I could be on track to graduate. And it turned out to be a wonderful experience. I learned all about insects, which I found to be really fascinating. And more importantly, I had a professor that took an interest in me. He invited me to do summer research with him, and that experience led to a publication and led to a career change for me. Instead of wanting to go to medical school, I wanted to go to graduate school and continue to study insects and hopefully eventually become a professor where I could then mentor students myself. So why did I fall in love with insects? Well, there's 3 big reasons for that. The first is about their biodiversity. So the International Union for the Conservation of Nature released in 2015 a number of diversity for different animal species. And what you can see in this figure is that if you look at all the species of known animals, 73% of them are insects. If you look at some of the other groups here, like the vertebrates, this includes all of our fish, amphibians, reptiles, birds, and mammals, they make up only 5% of the known animal species. So this is the group that we build all the aquariums and zoos to house and show off. Other animals like spiders, crabs, squids, and snails, and other invertebrates make up the rest. So insects are the most diverse group of animals on the planet. Second, insects are extremely important. There's lots of different ways they play a role for us. First, in terms of ecology, insects are important for the food web and for recycling. So this picture here has burying beetles that are disposing of a carcass. Insects also provide insights into genetics. For example, we use the fruit fly as a simple genetics model to understand more about our own genetics. In terms of biomedicine, insects are important disease vectors, like this mosquito for transmitting malaria or Zika virus. And finally, another way that they impact us is through food security. So pollinators are estimated to provide about $29 billion to the farming industry each year in terms of helping with crops. We also have crop pests, like this plague of locusts here, which can decimate the crops as well. So insects play a lot of important roles for us as humans. So probably the most interesting thing for me is their bizarre physiology. Let's take respiration, for example. So when we breathe, humans, we take air in through our mouth. It travels down into our lungs. It goes into smaller and smaller parts of the lungs called the alveoli. Oxygen then diffuses across the alveoli into the red blood cells, and the heart pumps the blood, which transports those red blood cells to the tissues, where oxygen is delivered, CO2 and waste products are picked up, and then the CO2 is transported back to the lungs, and we exhale. If we look at how efficient this system is, we can think about an elite athlete, like a cyclist, and we can measure what's called their maximal oxygen consumption. So this is the most oxygen they can consume at any one point. And if we look at an athlete like this, they consume about 5 milliliters of oxygen per gram hour. So this is the oxygen delivery system working at its peak efficiency and its top level. Now let's think about how an insect breathes. So insects, like this grasshopper here, don't breathe through their mouth, but they breathe through holes along their body wall called spiracles. There's one set of holes on each body segment. Oxygen and air enters these spiracles. It travels through an air-filled tracheal system. It gets into smaller and smaller branches of this tracheal system called tracheoles that delivers oxygen directly to the tissues. And then CO2 leaves through the tracheal system. But in no case do they use their circulatory system, blood, to transport oxygen. So it looks very bizarre and very primitive to us. But when we look at, say, a flying honeybee and measure its maximal oxygen consumption, we see that it's over 12 times the rate of our human cyclist. So even though it looks very bizarre and primitive, they're actually much better at delivering and utilizing oxygen than we are. So what does my lab at Union College work on? Well, we study these kinds of questions in insects using a variety of different methods. We do ecological work out in the field. We do organismal work in the lab. And we also study biochemistry of the proteins and enzymes inside the tissues. Today's work I'm going to be talking about was work I conducted with Jonathan Carroll, who is a former student. He's now currently a veterinarian student at Tufts University. And the overarching theme that my lab works on is understanding the mechanisms and consequences of biological trade-offs. So what do we mean when we talk about a trade-off? Well, we think about trade-offs every day in our lives. For example, tonight, perhaps, you have a trade-off to make. You could stay up late and continue to watch TV, or you could go to bed early and get up the next morning and exercise. So it's a trade-off you have to make. Now let's say you make the right choice and you go and exercise, right? Then you have another trade-off to make at the gym. Do you want to do endurance exercise, where you're running on a treadmill? If you do this continually for a long period of time, you're going to change your physique. You're going to develop into an athlete that looks more like this. Or you can make a choice, instead of doing endurance exercise, you can do resistance exercise. In this case, lifting weights. And if you do that for a long period of time, your physique will change and look like this. And we know, biologically speaking, what's happening at the level of the muscle. There's a different protein that gets turned on when you do endurance training, this PGC1 alpha 1. As this increases, it turns on things like the number of mitochondria that your muscle has. It increases the amount of capillarity, and your muscles end up becoming more oxidative. They're able to use oxygen better, and they show very little fatigue, so you can run for long periods of time, like this marathoner. Where if you're doing resistance training, you turn on a different protein, PGC1 alpha 4, and this turns on other signaling molecules, and your muscles go through hypertrophy. So they increase in size, so they're much stronger, but they don't deal well with oxygen, and therefore they fatigue more. So these are trade-offs that we see in an individual, that a human's sort of making. But what about evolutionary trade-offs that we see in the animal world? Well, one that's interesting, important to us, is this trade-off between gravidity, or pregnancy, and locomotion. So animals are going to want to produce the highest or greatest number of offspring for the next generation in order to be evolutionarily successful. But carrying eggs or carrying young comes with a cost. It increases the load that the individual has, and that load may affect the female's ability to get away from a predator. So there's a trade-off there. She wants to produce the highest number of offspring, but having too many could make her more susceptible to being eaten. And in fact, what we see in vertebrates is this case. So gravid sand lizards use shorter, slower escape runs when they're pregnant. We know that anolis lizards, which are made artificially heavier, about 30% of their body mass, they use shorter, less frequent jumps. And in mice, pregnant mice will run 85% less when pregnant compared to the non-pregnant mice. So we see a significant trade-off here in terms of pregnancy and locomotor ability. So the question our lab asked is, how does this trade-off affect things like in a terrestrial insect? Do they have the same trade-off that we see in the vertebrates? So to do this, we study Schistocerca americana, the American locust. And a locust is just a gregarious grasshopper. There's about 6,800 different species of grasshoppers in the world, and only 19 of them are classified as being able to be locusts. So a locust occurs when grasshoppers are in a high density. This high density leads to a change in the coloration and a change in their physiology and behaviors. They're more likely to swarm and migrate when they're in the locust form. So what we study is the Schistocerca americana, the American locust. These are native to the southeastern United States. Something really interesting about them is that when they develop from an egg, their first instar that hatches is about 15 milligrams. You can see that up here in the little arrow. And about 6 weeks later, they're an adult that's over 2,100 milligrams. So what that means in human terms would be if you had a baby that's born that's about 8 pounds, and 6 weeks later, it's going to be over 1,200 pounds. So there's an amazing amount of development and body size changes that occur in these animals. Another really interesting or wonderful aspect to these animals is that we can house them at Union College and rear them. So here's one of our cages in the vivarium. And these animals don't bite, they don't sting, so they work really well not only for research, but also for using in classes with the students at Union College. So once we had these grasshoppers, our first question was, how does body mass change with gravidity or pregnancy? And what we did is a student, Caitlin Arnson, measured body mass changes every day over a period of time. And we can see in the blue line here, these are the males, and you see that there's no difference in the body mass over these multiple weeks. But the females, on the other hand, go up and down during this time period. And what's happening is that they increase their mass by about 30% when they become pregnant every 4 days. And then on these red marks here are when they oviposit or lay their eggs. So for an animal of this female locust, it increases its body mass every 3 days, or every 4 days by 30%, and then lays its eggs. However, there's no compensation for its jumping muscle. So it has to deal with this increased load, but it doesn't increase the amount of muscle it might need to jump or get away from a predator. So we wanted to look a little bit more at what might be going on. So the first question we wanted to ask is, how does gravidity, or this egg carrying capacity, affect maximal jump distance? So what we did is we reared grasshoppers at 30 degrees centigrade. We selected for a range of body masses of both sexes, and then we clipped their wings so we can focus just on jumping and not flying, and isolated them for 4 hours so the handling wouldn't stress the animals out. Then we jumped the grasshoppers in a temperature-controlled room on a 5 centimeter by 5 centimeter grid. This is just basically a bed sheet. Knowing the numbers that they were jumping between, we could go back and calculate how far they jumped. So what did we find? Well, we found that maximal jump distance was not affected by gravidity. So body mass is on the x-axis here. Maximal jump distance is on the y-axis. The males are here and the females are in the green. And you notice there's some individual spread for each of these points, but in general, there's really no effect of body mass or gravidity on jumping performance. So this is the opposite of what we were seeing in the vertebrate data from the lizards and the mice that I talked about earlier. So if the distance they're jumping is constant, does the muscle performance change with gravidity? In other words, we know they don't have more muscle, but maybe the muscle of the gravid grasshoppers is doing something different. So John created a really interesting way to test this question. He built a system in which we could measure both the jump performance using a high-speed camera, as well as a muscle activity using electromyography, or EMG. And these were synced together with a trigger. This is what scientifically it looks like when we diagram it out. In reality, this is what it looked like in the lab. We've got, the jumping platform here, we've got a light, our high-speed camera, we've got a piece of a garbage bag here to provide some background so that we can videotape them. We've got wires strewn all over the place. You can even see John's bicycle in the background. So this is the part of science you don't get to see, what happens in the lab itself. What did we find out? Well, first we can look at the high-speed video. So the video is taken at about 500 frames per second. And you can see the video is running right here. The grasshopper is getting ready to take off. It's starting to take off. And the grasshopper has a mark on it so that we could track it through the video. So we could do analysis of the kinematics of the jumping. You can see it's trying to fly even though its wings have been clipped. Then I should add that clipping the wings doesn't affect the grasshoppers. It doesn't hurt them or stress them or harm them at all. So what can we do? When we go back and analyze these videos, we can find specific points on the video to look at. For example, we can look at their pre-jump flexing. We can look at their peak acceleration. We can look at their takeoff velocity as they're getting ready to leave. And we can look at their jump angle as they leave the ground. And for today's talk, I'm going to focus on takeoff velocity and jump angle. So what did we find? Well, we found that mass and gravidity did not affect the jump angle of the grasshoppers, nor did it affect the takeoff velocity. The data here is for takeoff velocity. And we've got the males, the non-gravid females, and the gravid females. And you can see, even though we have 8 animals per group, there's no differences among them. So if the jump performance as measured by a camera isn't any different, maybe there's something different going on within the muscle. And we can use electromyography or EMG to measure the muscle activity. So just like your heart has electrical activity, your muscle does as well. Every time you contract your muscles, an electrical signal changes. And we can measure that. For these grasshoppers, what we did is we anesthetized the grasshoppers with CO2, and then electrodes were implanted under the cuticle of the femur. So this is our jumping muscle here. And we put a ground electrode in the thorax. And we also did a sham control experiment. We went through the surgery and just to see if the effect of this surgery influenced their jumping ability, and it did not. So once we had the grasshoppers wired up, we could make them jump, and we could look at the electrical signal that the muscle was producing. So what did we find? Well, we found that these are individual EMG traces from different animals. And the way to read this is that the length of the trace indicates the length of the duration of the muscle contraction. So in the case of the male here, it's a pretty short EMG signal, so the muscle was contracted for a short period of time. As you get to the non-gravid female, it's a slightly longer, so they're contracting their muscles slightly longer. And for the gravid female, the EMG signal is much longer, so they're contracting their muscles for a longer duration than either the non-gravid females or the males. When we look at this across multiple individuals, we find some interesting relationships. So in this case, mass is on the x-axis. The contraction time of the jumping muscle, as measured with our EMG, is on the y-axis. And what we find is that as you go from a male to a non-gravid female to a gravid female, the contraction time increases. In other words, these gravid females are contracting their muscle longer than the non-gravid ones. And this is a significant relationship across all grasshoppers, and we also have a significant relationship just within the females. So from this, we can determine that the grasshoppers of different masses have similar takeoff velocities due to their increased muscle contraction times. So these gravid females are making up for the fact that they're carrying a much larger load by contracting their muscle longer, allowing them to produce a jump that looks very similar to the non-gravid or male animals. And this is an opposite of what we see in vertebrates, where an increase in load did affect their jumping performance. For the grasshoppers, increasing the load up to 30% did not affect their jumping performance. So why might this be? Why might grasshoppers be behaving differently than a vertebrate model? Well, grasshopper muscles aren't like our muscles. In fact, a grasshopper jumping muscle is a lot more like a catapult. So they're able to store energy in their jumping muscle. And what we think is going on here is that the gravid grasshoppers, like the catapult here, if you were to crank down on the catapult a little bit more, you're going to be able to store more energy and fire the rock farther, if you will. So the grasshoppers that are contracting their muscle longer are storing more energy and producing a jump that looks a lot like a normal or non-gravid grasshopper. This led to the question, is there a limit for this muscle compensation with an increasing amount of load. So we came up with another experiment where we looked at grasshoppers that were unloaded. We call them 0%. We looked at ones that had a weight attached to them that's equal to 20% of their body mass. The weight's on the back of their thorax. We looked at ones that had a weight that was equal to 40% of their body mass. And the question was, does adding this a weight affect their jump distance? And so what we found is on the x-axis here is the total weight. So this is the weight of the grasshopper plus the additional weight we might have put on them. And then the y-axis is their maximal jumping distance. For males, we find that during the addition of the weights, and these are for 3 individual animals, it doesn't really affect their maximal jumping too much. If we look at non-gravid females, we see that it affects it slightly, but again, not a huge difference in how far they can jump. But when we look at gravid females and add weights to them, we notice that the maximal jumping distance decreases significantly. So adding these weights to a gravid female that's already carrying a large load makes a significant difference. So what happens if we look at, say, the jump performance with our high-speed video system or the muscle activity with our EMGs? So we repeated our experiment. You can see the grasshopper getting ready to jump. It's got the weight on the back, and it also has the wires coming out for the EMG. And if we look at the jump performance first, measured with our high-speed video, we see that takeoff velocity was similar regardless of the amount of added weight. So here are the males in blue. Again, no real difference in takeoff velocity, whether they were unloaded, had a weight that was 20% of their body mass or 40%. For the non-gravid females, again, it drops off a little bit, but not statistically significant between the unloaded, the 20% of their body mass weight, and the 40% of their body mass weight. And for the gravid ones, again, it drops off slightly as you add more weight on them, but their takeoff velocities are not statistically different. So something's not happening with what we can see with the video, but something might be happening in the muscle itself. So if we go to do the EMG, what we find is really interesting. On the x-axis, again, is the total weight. The y-axis is the muscle contraction time. And the longer the muscle contracts, the more energy they're going to be able to store. And so I've just presented the female data here. And what we see is that for the squares, these are the non-gravid animals. So these solid squares are the unloaded animals. These squares that are colored with the pink are the 20% weight, and the open squares are the 40% weight added. And for these non-gravid females, you see that as you add weight to them, they increase the contraction time of the muscle, as measured with this EMG. And it's a significant relationship. For the gravid animals, these are the unloaded ones that are the full green circles. This grayish color is the 20%, and this is the gravid with a 40% weight. There's no significant difference across here about how long their muscles are contracting, regardless of how much weight you add to them. So what we can conclude from this data is that the gravid females are at their limit for muscle contraction duration. In other words, adding those weights to them did not increase the EMG signal or the muscle contraction time. Where the non-gravid ones, their added weights increased the EMG signal and allowed them to have a greater muscle contraction duration. What this also means is that we think gravid females have maximized this trade-off between carrying egg mass and their locomotory jumping performance. So the question that comes to mind probably is, why aren't all grasshoppers jumping maximally? If these non-gravid females, for example, could jump more, why aren't they producing longer jumps? Well, we think there's a couple possibilities. One is that this might be a way to reduce potential damage. If they're always jumping maximally, they're more likely to damage their muscle, a ligament, or a tendon, especially if they don't need to. Second, maybe what's important for them is their takeoff velocity necessary for flight. So if they can achieve that takeoff velocity without using a maximal jump or contract their muscles longer, then maybe that's the best thing. Or perhaps it allows them to jump more frequently. If they don't have to contract their muscles for as long, then they can jump more often, especially useful for getting away from predators. This last point about jumping frequency has led us to our new avenue of research. So we're interested in how gravidity might affect repeated jumping, which is thought to be an aerobic activity where oxygen delivery is important. And to study the oxygen delivery side of this, we're using our new nationally science-funded microcomputed tomography system. So we acquired this through an MRI grant that I wrote. And this system works a lot like a CAT scan. It allows us to get resolutions down to less than a micron. So here's a picture of a grasshopper femur. So the head of the femur is here, and this is in a longitudinal view. And what the system does is it takes a series of X-rays, slices, and here's one X-ray cross-section slice. The openings here in black are the tracheal respiratory tubes that are delivering the oxygen to the tissues. And one can go through the software associated with this system, and you can stack these images together and create a volumetric representation of the tracheal system, as indicated in blue here, and the green here indicates the rest of the surrounding tissue. So what we want to do is measure the volume of the oxygen delivery system in gravid and non-gravid grasshoppers throughout their entire body, and eventually create reproductions that look like this. So this would be the thorax of the grasshopper, this would be the abdomen of the grasshopper, this is the jumping leg here, and everything that you're seeing there is the tracheal system. So that way we can look at how oxygen delivery systems compare in non-gravid and gravid grasshoppers. So the importance of this work comes back to the role locusts play in the world. So a locust swarm can have up to 80 million individuals. And locusts have an amazing impact. In fact, they're oftentimes the most economically destructive pest insect on a continent. For example, the African desert plague locust in 2003 caused $2.5 billion in damage and required $450 million to stop the outbreak. Locusts feed on many cereal crops, which are important crops for global food security. And in a plague year, locusts can impact 1 in 10 people and cover 20% of the Earth's land surface. So these facts come from the Global Locust Initiative, which is a new group out of Arizona State University organized by Dr. Ariana Cease. And this group brings together researchers, it brings together government and non-government officials, and it brings together farmers that have all been touched by locusts around the world to try to come up with ways to reduce the devastation of these insects. So we're hoping some of our research will help at least provide some insight in how locusts not only jump, get around, but in terms of the amount of eggs that they can produce. Finally, I want to make some acknowledgments. I want to thank Union College that supported this research through the Faculty Research Fund, through the Student Intellectual Enrichment Grants that allow my research students to spend the summers working with me or traveling to national meetings to present their data, and also the biology department that has helped fund this. And a lot of those funding sources get their money from the alumni that donate. So thank you too for sending donations to Union College. It helps what we're doing in our research labs. I also want to thank the National Science Foundation for funding this research, especially the micro-CT, and John Carroll, who I worked with to collect and analyze this data. John is just one of the many wonderful students I've had the opportunity to work with over the years that has made this part of my life incredibly enriching. So I want to thank you for listening, and I'm willing and happy to answer any questions about this research or other work that my lab does. Thank you.
Thank you, Professor Kirkton, for that wonderful and engaging presentation. If you have a question for Professor Kirkton, you can email us at alumni@union.edu. Professor Kirkton, we have a couple questions for you while we're waiting for some folks to email their questions. Mm-hmm. You said that insects that have more species than all other animal groups put together. How many different species of insects are there, and why are insects so evolutionarily successful?
Oh, that's a great question, Phil. I'm glad you asked that. So there are estimated to be over 1 million different species of insects on the planet. Every year, about 9,000 new species of insects are discovered. And what's really interesting, if you look at the diversity of the species, are that there are only about 15,000 known species of birds. But beetles, which are just one type of insect, there are 350,000 different species of beetles. Besides just the species diversity, it's been estimated that there's 1.5 billion individual insects on the planet for every human being. So that's a lot of bugs.
Why are there so many different species?
Well, it's a good follow-up. They're thought to be about 4 to 6 million species of insects on the Earth, but the rest haven't yet been discovered. Why are they so diverse? Well, there's a lot of reasons for that. A couple of them include their small size. So they're able to exist in lots of microhabitats that are off limits from vertebrates. For example, one tree could house 42 different species of ants in the Peruvian rainforest. Another aspect is there are different types of ways they feed. So an acacia tree found in Africa can support a giraffe, which eats its leaves. Insects have lots of different feeding guilds. So there are seed eaters, there's leaf eaters, there's wood borers, there's ones that eat the roots, there's sap feeders, then there's parasitoid wasps that will come feed on those herbivorous insects. There's also predatory insects. The tracheal system, which I talked a little bit about in my presentation, it allows them to have really high oxygen delivery systems, so they're very energetically active. Insects also have an exoskeleton, this armored protection that allows them to be well protected as they're traveling through the environment. They have wings so they can migrate. They have quick generation times. So while vertebrates have to learn about their environment and adapt that way, insects, their populations can turn over quickly and they can evolve based on, say, environmental changes. Also, metamorphosis is another important thing. So it separates out, say, the juvenile, like a caterpillar, which feeds on plants, from the butterfly, which feeds on nectar. And so lots of insects go through these metamorphosis or stages that allows them to separate out different food products. It also allows them to get through extreme environmental problems, like a caterpillar that becomes a cocoon and then can deal with this extreme winter in that cocoon stage. So there's lots of different ways that insects have evolved. They also have lots of co-evolution with plants. So as you see, like a milkweed is linked to the butterflies that feed on the milkweed. So we have certain links there, coevolution between plants, and there's coevolution between different insects as well.
Okay. Again, you're watching Chet Chats with Professor Scott Kirkton of Biological Sciences. If you have a question for Professor Kirkton, you can email us at alumni@union.edu. Professor Kirkton, what advice would you have for a young person who may be interested in studying biological sciences?
Oh, that's a good question. So I think there's some inherent traits that a scientist should have. They should be intellectually curious about the world around them, about how things work. They should have some ability to deal with frustration and failure. So I had one mentor that told me, in order to be a good scientist, you have to be happy and excited by the 10% of the time that your experiments work, as opposed to the 90% of the time that it fails. There's obviously some skills that you need to have and be aware of. Mathematics, biology, chemistry, physics, engineering, those are all parts that play a role in my research, having some knowledge of those. But also the liberal arts can contribute a lot to a scientist. Creativity, which is often thought to be just the realm of humanities and arts, is really important in science. So being able to creatively think about experiments, designing experiments, as well as knowledgeable about how things work in the liberal arts. For example, I learned a lot coming from a liberal arts college about, for example, how to deconstruct a poem or a piece of literature. And the ability to deconstruct and break something apart are the same skills I use in my scientific laboratory. So a lot of those skills overlap. I think we sometimes think of them as being science and engineering over here, and liberal arts, humanities, and social sciences over there. But the skill sets are very dependent on each other.
And what opportunities would a student have at a place like Union?
Well, at Union, students who are interested in doing research or getting some hands-on experience, contacting a professor is the best way to get started. So I have had lots of first-year students working in my lab. Sometimes that's an experience that they really enjoy, and they continue with that throughout their 4 years, and they get to attend scientific conferences, write a thesis, write a paper, hopefully get it published. Sometimes they experience that and they realize that maybe that's not what they want to do. And that's a good thing to know too, right? Maybe they want to do something else with their career. But my lab has students from all different grades, seniors, juniors, sophomores, and first-year students. And we try to create kind of a mentorship system where there's upperclassmen mentoring some of the underclassmen, so they're working together. I think the hardest thing for students getting a chance to work in a science lab at a place like Union is that they're so used to their scientific classes at Union where they're doing the lab components, where those labs have all pretty well been thought out. We kind of know what's going to happen. And when you get to start doing original research, like I said earlier, you're not sure what's going to happen. And you have to deal with those problems that arise. And so having a student that's willing and able to deal with all the failure that you're going to be faced and having to repeat things over and over again until it succeeds, they have to enjoy that because they're contributing new knowledge to the scientific world.
Okay. Again, you're watching Chet Chats with Professor Scott Kirkton of the Biological Sciences. If you have a question for Professor Kirkton, please email us at alumni@union.edu. We do have a question for Professor Kirkton. Since pregnant mice show reduced running ability, do pregnant women also show reduced exercise capacity?
Oh, okay. So that's an interesting question. The field's pretty diverse in kind of the responses for things like that. What's known is that for pregnant women, when they are exercising with weight-bearing exercise, so walking, jogging, running, that the oxygen consumption rate goes up compared to a non-pregnant woman, and that's due to the extra weight they're carrying. And this changes during the course of the pregnancy. So that causes them to have a sort of a lower aerobic capacity or needing more oxygen in order to carry out the same activity. So there are also ligament changes and other biomechanical changes that occur in terms of the joints with pregnancy. What's interesting is if pregnant women are doing cycling, for example, non-weight-bearing activity, that their exercise capacity is the same.
Interesting, interesting. You talked about some of the opportunities that a student would have at a place like Union. Can you share some of the other research your lab is working on or has worked on?
Oh, sure. So this project I talked about today is just for one aspect of the research that we think about in my lab. As I mentioned in the talk, the animal model that we use, Schistocerca americana, the American locust, goes through this change in body size. So it hatches as a 15 milligram individual first instar and then becomes over 2,100 milligram adult, so in about 6 or 7 weeks. So it's a dramatic change in body size. And so one thing we've been interested in my lab is looking at those changes in development. So if you're increasing your body mass that dramatically, how does that affect your physiological systems? So some of the things we've been looking at are the jumping performance, the oxygen delivery system, as well as the muscle performance. And what we found has been pretty interesting. So these little juveniles, when they hatch, they are much more aerobic in terms of their exercise and activity. So they can jump and jump and jump without fatiguing. So we sort of refer to them as the marathoners. And as they go through different stages, they become more anaerobic. And the adults actually produce the most anaerobic lactic acid. So it's the same molecule that we produce when we're sprinting. So they go from being these marathoners to these sprinters during this course of development. So we're really interested in how that's happening and why that's happening. We've taken these grasshoppers to Argonne National Labs outside Chicago, where there's an X-ray synchrotron that we can X-ray the images of the animals while they're living, and we can measure their tracheal system. So Union students and myself, we've traveled there in collaboration with people from North Dakota State and Arizona State University. And what we found is that the larger older grasshoppers actually dedicate more of their body tissues to the respiratory system. So suggesting that they might have more problems with oxygen delivery, hence the lactic acid they produce in their muscles, or the fact that when you x-ray them, they've got more trachea per body mass than the smaller ones. And the reason why this is really cool is that there's this hypothesis that's been around for a long time that Paleozoic gigantic insects existed because of oxygen differences in the atmosphere. So about 250 million years ago, in the late Paleozoic, there were giant insects, hawk-sized dragonflies. And so fossil evidence shows that these large insects existed at a time when oxygen levels in the atmosphere were about twice what they are now. And so the thought was larger insects existed because it was easier for them to breathe and get oxygen. And so what we've kind of seen in this developmental study here at Union is that these larger grasshoppers are having, at least in some cases, having more problems breathing So it provides a little bit of support for this idea that, yes, maybe these large insects occurred because oxygen was more readily available for them.
Okay.
So that's one project, and there's, we're doing other neurobiology studies where we're electrically stimulating the muscles, like training them to see if we can change muscle fiber types. We've done studies, preliminary work, where we go out to the field in the Adirondacks and look at native grasshopper populations from high and low altitudes and trying to see if any evolved differences have occurred in terms of their oxygen delivery system and jumping performance. And we've been looking at changes within an instar. So as I mentioned, these grasshoppers go from first instars up to adults, and they go through 6 developmental steps. And each one of these steps is about a week long. And at the end of each step, they have a molt, so kind of like a snake shedding its skin. And so we're interested in what happens during that week period, where at the beginning of the molt, you have this exoskeleton that's not going to change. By the end of the molt period, you've increased your body mass. So is that increase in body mass while you're still contained in the same form, how does that affect as you're compressing maybe these air-filled tracheal tubes? So we're looking at sort of, maybe you can get away from a predator at the beginning of the week, but by the end of the week, you can't. You'll be dinner.
Wow. Fascinating stuff. You've been watching Chet Chats with Professor Scott Kirkton. Professor Kirkton, we'd like to thank you for sharing your expertise with our at-large union community. We appreciate
Thanks, Phil.
that.
I'd like to thank Phil Wida for agreeing to sit here with me and ask the questions. Rhonda Engvold from the Alumni Relations Department, and then the Information Technology Services Group at Union, which includes Jermaine Wells, Jim Strohecker, and Bijit Sarkar, who helped make this presentation possible. So thank you.
Thank you.